ICSE Concise Biology, Chapter 2, Part 2, Section 2.8, Cell Division. Cell division is of two types, mitosis and meiosis. Mark the two definitions. In mitosis, the parent cell divides to produce two daughter cells which are diploid, meaning they have the same number of chromosomes as the parent cell. So if the parent had 46 chromosomes, then each daughter cell will also have 46 chromosomes. That is because during the cell division, the DNA is duplicated, so that totally there are 92 chromatids in the 46 chromosomes which get separated during cell division. Whereas in meiosis, the duplication of DNA doesn't take place. The number of chromosomes are halved in the daughter cells. That takes place only in the gametes formation, that is sperms and egg cells formations in the reproductive organs of the body. And the cells are called haploid cells, not diploid cells, which is represented like this. So every cell of a body has 46 chromosomes, 23 pairs, 23 from your mom and 23 from your dad. 2n, diploid condition. But when the sex cells are produced, that is the sperms in the testes or the egg cells ova in the ovaries of a female, each daughter cell will have 23 chromosomes, so they are halved. That's important so that when they will fertilize and a zygote is formed, the original number is restored, 46 chromosomes. We don't want the sperms to have 46 chromosomes and the egg cells to have 46 chromosomes, because if they fertilize, then the zygote will have 92 chromosomes, which will not grow as a human being. Maybe it will become a Godzilla, which your parents wouldn't want. Now let's study the different phases of mitosis. There are two main stages, karyokinesis and cytokinesis, which we studied in the cell cycle. Karyokinesis is the division of the nucleus, and then the cytoplasm divides, which is called cytokinesis, to give birth to two daughter cells and the parent disappears. Now let's study the various phases inside karyokinesis, and the best way to study this is with the help of diagrams. So double tick figure 2.10, practice all the pink diagrams. Figure 2.10, double tick it. The plant cells need not be practiced, although they can also be asked in exams, but they are very similar. So the theory part, you can mark it as read. So this will be read, metaphase. So after prophase comes metaphase, read, anaphase, read, and even the telophase, read, because we are going to study from the diagram. So first we have the interphase. The interphase is not called a resting phase. Cancel resting stage from here and here. If you remember the cell cycle, the interphase comprises of three steps, G1, S and G2. If the cell does not wish to continue with the cell cycle, then it will go into the resting phase. So interphase is not called the resting stage. In fact, a lot of things are happening in the interphase. It is not resting. But under a microscope, there are no visible changes. DNA is being duplicated during the synthesis phase. We see the chromatin threads inside the nucleus, nucleolus, nuclear membrane, centrosomes in animal cells, but not in plant cells. And now the mitosis starts, and the first step is prophase, early and late. In exam, when they give you a diagram and ask you to identify the phase, you don't have to mention early and late, you can just mention prophase. What are the characteristics of a prophase? Well, you can see the centrioles are duplicated and they move apart to opposite poles. Spindles are starting to be formed between them, spindle fibers. The chromatin fibers condensed and organized into chromosomes. The sister chromatids have been formed, which are attached to each other at the centromere. The nuclear membrane is disappearing. In the plant cells, there are no centrioles, but there are spindle fibers made from the microtubules which were present in the cytoplasm itself. Next, we have the metaphase. It's quite unique because only in this phase do we see the chromosomes lined up vertically along the equator of the cell. And the spindle fibers are attached to the centromere, ready to pull apart the sister chromatids, which happens in the anaphase. The sister chromatids move towards the opposite poles because of the contraction of the spindle fibers. Notice that the asters also exist in the animal cells. There are no asters or star ray-like structures in plant cells. So if this was a human cell, 46 chromatids will go left and 46 chromatids will go right, so that each daughter cell has the same number of chromatids or chromosomes. So So here, how many chromosomes are seen? I can see 4 chromosomes, and there are 8 chromatids because each chromosome has duplicate chromatids called sister chromatids. Now, I also see the furrow formation has started here. And in the last phase of karyokinesis, telophase, the furrow deepens, the cleavage deepens, and the chromosomes again become chromatin fibers. The nuclear membrane reappears, the nucleolus reappears, the centrosomes are seen, the spindle fibers disappear. In the plant cells, there is no furrow formation. Instead, the cell plate starts to form, and that's the beginning of cytokinesis. The karyokinesis has ended, but the two nuclei share one cytoplasm which needs to be divided, and so two daughter cells are formed. And here in the plant cells, a cell wall is formed in between. Now mark the definition of karyokinesis and cytokinesis. Why are the daughter cells called so? Why not son cells? Well, because those cells are further able to divide and reproduce. Hence, it's just a convention. Now, what's the difference between mitosis in plant cell and in animal cell? Here, asters are formed, asters aren't formed. Furrowing happens, cell plate formation happens. This happens in almost all parts of the body. Mitosis does not happen in the reproductive cells, by the way. This happens in plants only in the meristematic tissue, but the permanent tissues don't divide. Spindles are formed with the help of the asters and the centrioles, whereas here they are formed with the help of microtubules. Now, if a diagram is given and you have to identify the stage, then you can look at the characteristics to state what is happening. In the first diagram here, this one, it's just an interphase because I see a single nucleus and chromatin fibers, no chromosomes. In number 2, chromosomes are being formed, although not seen clearly, but this is prophase. How do I know that? Because that is what is mentioned here. Third one is clearly metaphase because all the chromosomes are in the middle of the cell, ready to be split. Fourth one is the anaphase because the splitting has occurred. Fifth one is telophase because now two nuclei have been formed and a cell plate is being formed. And sixth one is finally the end of cytokinesis and two daughter cells formed with two tiny nuclei which are a duplicate of the original nucleus. So what is the significance or the importance of the mitosis? Well, they are important for growth and repair and replacement of cells. Can mitosis be useful in reproduction? Yes, in asexual reproduction. For example, binary fission in bacteria. It happens by mitosis. There is no meiosis and fertilization and zygote formation in bacteria or amoeba. And mitosis is important to maintain the same number of chromosomes as the parents in the daughter cells. If you remember, in the G1 phase of interphase, the mitochondria and chloroplasts multiply after mitosis. They can multiply on their own because they have their own DNA and own ribosomes. They are semi-independent. Now meiosis. If mitosis was called equational division, meiosis is called reduction division because here the number of chromosomes are halved and it takes place only in the testis and ovary of animals and in the anthers and ovary of plants to produce gametes. Now mark the give reason. It's called reduction division because number of chromosomes are halved in the daughter cells. From a diploid condition, it converts to a haploid condition. Now this diagram is not in syllabus, but let's just understand it. So this parent cell has 3 pairs of chromosomes, 6 chromosomes. When it undergoes meiosis, each daughter cell will have only 3 chromosomes. But notice that unlike mitosis, the daughter cells are not identical. Here I can see 2 yellow chromosomes from the mother and 1 green originally from the father. But here we have 1 yellow and 2 green. So they are not identical, which has some significance I'll explain later. But that's not the end of meiosis. That's just the first reduction division. Then we have the second mitotic division. The identical daughter cells are formed from them. So ultimately, 4 daughter cells are produced. Notice that these two are identical and these two are identical to each other. Now in this paragraph, the last sentence is just read. Significance of meiosis. Importance. First one is a GR. It is important in producing gametes because the number of chromosomes are halved, which is important so that when fertilization takes place, the original number is restored. But meiosis is important in one more way. It helps to mix up the genes. It helps to create variations in the offspring. Why is it that even if the same couple have multiple children, each child is unique? Now, all of them get chromosomes from the same parents. Then how come each child is different from the other? To understand that, let's study a bit of chapter 3, genetics, figure 3.2, page 25. If you zoom into the nucleus of a dividing human cell, you will see 23 pairs of homologous chromosomes. Homologous chromosomes are the ones which have the same shape and size, they look similar to each other, and they have information about the same characteristics. So in each pair, one of them is originally from the mother and the other is from the father. Now let's imagine that the first chromosome has information about the color of your iris. So let's say this has information of blue iris and this has information about brown iris in the form of genes in the DNA in the language of ATGC. And the second chromosome pair has information about your height. Let's say the first chromosome has information about tallness and the second is about short-heightedness, dwarfness. Now when the chromosomes divide, each sperm or each egg cell will get only 23 chromosomes out of it, not all 46 chromosomes. So from each homologous pair, only one of them will go into a particular sperm. So the sperm will not have both the informations of blue iris and brown iris. It will have only one of them. So some sperms will have information of blue iris and some will have information of brown iris. Similarly, some sperms will have information of tallness and some of shortness. So there are thousands of such characteristics, and so there will be so many permutations and combinations in the sperms. Each sperm is unique. Some sperms will have a combo of blue iris with tallness, and some sperms with brown iris with tallness, or brown iris with shortness, and so on. Now, which sperm will fertilize which egg cell is just purely a matter of chance. So, if the child will have iris like the mother, or iris like the father, or like the grandfather, and will it be tall or short? It all depends on this fertilization chance. So many combinations are possible. Even if both the parents have brown iris, it's possible that the child will have blue iris. So don't have suspicions why the child has blue iris, even though the parents have brown iris. It's possible that the parents did have the blue iris information hidden. It was suppressed because brown is dominant. Maybe the grandparents had blue iris, which skipped a generation, as now is seen in the children. All of this will be explained in detail in the chapter number 3. But now you know why meiosis is important. It creates new combinations which were not even present in the parents. But not only that, that's only half the story. Now, what if in a homologous chromosome, figure number 2.12, single ticket, the homologous chromosomes have the same information, that is blue iris and brown iris, and tallness and shortness is also in the same pair. Now you may think that if this chromosome goes into the sperm, then the blue iris and the tallness information will be together. But that's not the case. While they are close to each other during cell division, meiosis, the part of the chromatid may exchange with each other. This phenomenon is called crossing over, and this X-like link is called chiasma. Mark the definitions. So genetic recombination is happening. Now we have a chromosome which has information of blue iris and shortness, something which might not have existed in the parents or the grandparents. A new combination is created, and that is why new varieties are born. And variations are good for the evolution and the survival of the species. We don't want all the species population to be exactly similar to each other. So meiosis helps in creating variations in two ways. First, it mixes up the maternal and paternal chromosomes in the homologous pairs. And second, the crossing over helps in genetic recombination, creating new varieties. And the crossing over happens between the non-sister chromatids. The yellow were sisters, the pink were sisters, but the yellow and the pink are non-sisters. They belong to different chromosomes. They are a homologous pair, one of the 23 pairs in the human cell. Now differentiate between mitosis and meiosis. Mitosis happens in all the body cells, the somatic cells. Meiosis happens only in the reproductive cells. This happens for growth, repair, replacement. This happens for gamete formation. This happens throughout our life. This happens only during the reproductive age. It starts at around, say, the age of 10 or 12 and lasts till you are fertile. This results in the formation of 2 daughter cells. This will ultimately form 4 daughter cells. Here the daughter cells will be diploid in the end. Here they will be haploid. A single nuclear division happens, 2 nuclear divisions happen, a reduction division and a mitotic division. The daughter cells are identical, copies of each other. But here the daughter cells are different because of the random assortment of the gametes, of the chromosomes in the gametes. There is genetic variation between the gametes. Each sperm is unique. And out of the millions of sperms, you won the race and fertilized the egg cell and became the child. You were a winner even before you were born.
Hi students, this is AJ sir. If you found this video useful, press the like button. Also, if you'd like to enroll for my test series or online lectures, email me or message me on Instagram. Check my description for more information.